So, students, now we are going to move toward that microbes which are used in industrial productions. In last video, we learned about that microorganisms which are used in household productions. Okay, so that microorganisms which are used in household production. So now we are going to move toward that microorganisms which are used in industrial productions. At the level of industrial point of view, or at the industrial level, the different products which are produced is in large scale. To produce that product is in large scale, so that some kind of microorganism which are used, and that microorganism so which are used to prepare somewhat the industrial products, somewhat the industrial products. That industrial products which include, first, that is nothing but you can see it as a alcoholic beverages. In Marathi we call it as a पेय. मद्यपेय वगैरे तयार करण्यासाठी या ठिकाणी सूक्ष्मजीवांचा वापर होतो. इंडस्ट्रियल प्रोडक्ट्स लाईक अल्कोहोलिक बेवरेजेस. Alcoholic beverages not only used for the drinking purpose, they are also useful for the medicinal purpose. So to prepare the sanitizer, different kinds of, we can say it as perfume, even also they are useful to prepare the different kinds of, we can say it as surface cleaners. Okay, so here we just take that industrial products. What about the alcoholic beverages? Different kinds of antibiotics, various kinds of enzymes, different hormones, just like gibberellins, different kinds of hormones, some kind of bioactive molecules, etc. So which are produced at the industrial level. Not only all these, but also different kinds of other chemicals, just like some kind of enzymes, that vitamins, that is nothing but also some kind of antibiotics. So at the large extended stage we can easily prepare by using that different microbes. So now we just move toward that preparation of alcoholic beverages. Alcoholic beverages which include, we can say it as the different alcoholic beverages for using Saccharomyces cerevisiae. The name of species used which is somewhat same, that is Saccharomyces cerevisiae, but the products are different as per the substrate. Different alcoholic beverages formed using Saccharomyces cerevisiae, but different products based on substrates. Okay, so here we just take, that is nothing but we can say it as a, first of all, that is beer, second, that is wine, third, whiskey, So beer which is prepared from barley, wine which is from the fruit juices, this is from mixed grain, this is from potato extract, okay. So out of them above two, so they are non-distillated product, non-distillated, which means nothing but after the fermentation, so they are not required the heating, evaporation is not required for the these upper two products. What about the beer and wine? And so that's why we call them as a non-distillated. But the remaining two, after the fermentation, they require the heating, and after heating, they can be purified or separated. Okay, and so that's why we call them as a distilled product. Okay, distillated product. They are said to be the distillated products. So here we just take that preparation steps for the preparation of beer, steps for beer formation, This point is asked for the board and CET. So here steps for beer formation. First of all, that is nothing but the separation. We call them as a malting process. Separation of grain, that is said to be the malting. Second one, that is nothing but the mashing process. After the isolation of grains, that grains which are crushed out easily, we can say it as a hot water, and so that's why we call them as a mashing. Then that is fermentation. After fermentation, that is nothing but clarification, and last one, that is carboxylation. So these are the stages for the beer production. Okay, so like that. So they are the different alcoholic beverages they have developed is in the industrial level. Okay, so just keep in mind that are the things. So students, now we are going to move toward what about the antibiotics. So the term antibiotics, which is composed from two words. So the first word, that is nothing but the anti, that the word anti, which means nothing but the against, and the term bio, which is related with the living organism. So it means nothing but that antibiotic means nothing but the against the life. But this term is somewhat misnomeric, because so antibiotics are not against the life. They are very important, they are crucial, essential for us. Because today, without antibiotic, so that word is not imagined. Okay, and so that's why the proper definition of antibiotic is a chemical produced by a microbes, a chemical produced by a microbe to kill or stop growth of other pathogenic microbes. This is the definition for the antibiotics. It is a chemical which is produced by a microbes to kill or to stop the growth of other microorganisms which are somewhat pathogenic to us. Okay, so here we just take that antibiotics which are very important to treat the many diseases. So here we just going to move toward how that first antibiotic was discovered, who discovered that first antibiotic, and also about that how that antibiotic was prepared is at industrial level. Okay, so just see here what about the first antibiotic. First antibiotic coincidentally discovered, that is said to be the penicillin. First antibiotic, so first antibiotics, so penicillin coincidentally discovered by Dr. Alexander Fleming. Dr. Alexander Fleming, when he was working on first antibiotic penicillin, that is Dr. Alexander Fleming discovered when he was discovered, he was work on, sorry, when he was working on, staphylococci or staphylococcus. We know that the staphylococcus, that is coccus, cocci, that is nothing but a rounded spherical bacterium which are somewhat like that. So spherical bacterium are called as cocci, and here that cocci, if they are found in the straight chain, we call them as streptococcus. But here, if that bacteria which are found in branch chain, which are found in such a kind of branch chain, so we call them as staphylococci. So actually, so that Alexander Fleming cultivating that staphylococci is in petri plate, okay, and he just conducting experiment on the staphylococci and observe that staphylococci not growing here, where that actually that Penicillium notatum is found. Penicillium notatum, the name of fungus. So here that is somewhat Penicillium notatum. Penicillium notatum is found in this area. So here no one that staphylococci colony formed, and so that's why here we just observe that Penicillium notatum which produces such a kind of chemical which inhibit the growth of streptococci or staphylococci. So that's why we just call that penicillin, which is an antibiotic, work against bacterium. Okay, and these antibiotics, so it was used extensively in Second World War to treat or to heal wound of American soldier. After that, two scientists, we call them as Ernst Chain and two scientists, that is nothing but the Ernst Chain and Howard Florey. more properly discovered its use. Okay, like that. So that is about the antibiotic. Recently, so many antibiotics prepared from that streptomyces, streptomyces used to prepare, many antibiotics, just like erythromycin, streptomycin, okay, etc. Actually, uses of antibiotics in plague, whooping cough. In Marathi we call it as a kali khasi. Number third one, it is also useful to treat diphtheria. We also call it as gulgotu. Number next, plague, whooping cough, diphtheria, and not only is in old diseases, but also it is useful to treat is in many recent diseases, even that is sore throat, etc. Okay, so this is about, we can say it as the term antibiotics, and the introduction about the antibiotics is at industrial level. Okay, so just keep in mind that all things what about the antibiotic. So after that we will going to learn about that next part, that is said to be the production of enzymes and different bioactive molecules.